Hi, everyone. Welcome to the Vernal Pool Phenology Project information session. I'm Mary Beth. Before we get started, I just want to give a little overview of the format. It's probably going to be about 20 minutes of us talking, but we'll have a couple opportunities for open questions and answers. We'll start off by just introducing the project, our overall research question, what you need to participate in the project. Then we'll take a little break, have some questions and answer time, and then launch into the sampling protocols and introduce the teaching materials that we have available for your use. And then we'll go into the second round of questions and answers. So feel free at any point to ask questions using the chat feature. We'll try to respond. We'll be monitoring it. We'll try to respond if we can right away. And also feel free to ask your questions during the open question and answering times. The info session is being recorded and we'll be posting it to our YouTube channel. And we'll also be following up this session with a survey that we should be sending out to you next week. To get a little bit more information. But I do want to remind you that at any point that you have any questions after the session or whatever, feel free to email us at our project Gmail, which we'll be showing you later. Next slide. So before we get started, we want to introduce the Vernal Pool Phenology Project leaders, as well as acknowledge the 2022 Vernal Pool Phenology Project Pilot 1 year participants, Lori, Mike, and Tim. They were instrumental in helping us develop our protocols and testing our first round of protocols so that we could open up this to a much larger audience for pilot season 2.0 in 2023. I'm Mary Beth Kolesrari. I'm an associate professor of environmental studies and sciences at Siena College, which is right outside of Albany, New York. Dave?
I'm Dave Steinberg. I'm a lecturer at the University of New Hampshire in Durham, New Hampshire. Jen?
Hi, I'm Jennifer Pernhage, and I'm a senior lecturer in wildlife and conservation biology at UNH.
Hi, I am Thilina Surasinghe, Associate Professor, Biological Sciences, Bridgewater State University, Mass.
Next slide. So before we launch into the Vernal Pool Phenology Project, we thought it was important to introduce the Ecological Research as Education Network because the Vernal Pool Phenology Project is an EREN project. So first off, EREN has been a network that has been in existence since 2010, initially funded by NSF through a research coordination network grant to help get us up and running. To describe EREN, I'm going to read the mission: to create, facilitate, and promote collaborative ecological research that generates high-quality publishable data involving undergraduate students and faculty at primarily undergraduate institutions or PUIs. So EREN works to bring big science to small institutions. I think our mission statement really captures what EREN is all about. I'll just mention a few things about PUI faculty. PUI faculty have research expectations, but we also have very heavy teaching loads. And we're expected to engage undergraduates in meaningful, authentic, hands-on ecological research, both in the classroom, in the field, and as independent studies. So PUI faculty are really, the EREN network really has allowed PUI faculty to be able to work together addressing large-scale ecological questions at the regional and continental scale, using standardized protocols, hands-on with the undergraduates and faculty, hands-on collecting those data, analyzing those data, and contributing to be able to address these large-scale ecological questions. Another thing about PUI faculty is that many of our institutions don't have a lot of funding and support to purchase expensive research equipment. So EREN keeps all that in mind. They recognize the importance of engaging undergraduates in hands-on ecological research. They recognize that the limitations or the other needs on PUI faculty. And so we try to make it easy for faculty to be able to bring these research questions and projects into their classroom. EREN projects are led by lead scientists. They're proposed and led by lead scientists. Lead scientists are responsible for providing detailed protocols, teaching materials, information, sometimes like videos or whatever it can be to help make it easy and engaging to encourage participation by PUI faculty in these research projects. And again, remember the end goal is to have high quality publishable research involving both undergraduates and faculty at PUI institutions. Next slide. And we're a very inclusive, supportive network. We are not just PUI faculty. We have a lot of R1 institution faculty that are lecturers, that have more teaching experiences. It doesn't matter. Why we emphasize the PUI faculty is we recognize the unique nature and role of them, and we make sure that the research projects are designed with them in mind so that they're able to participate. But we're very inclusive. We have had high school teachers and students involved in projects. So whether or not you're at an institution or not, our projects, we welcome and encourage your participation. Next slide. Now to introduce the overall research question for the amphibian and plant phenology at Vernal Pools project. What we're designed to do is address how do local and regional factors affect the spring phenology of pool breeding amphibians and trees. So most of us are probably already familiar with vernal pools. So let's just think about vernal pools themselves, the fauna that exploit these seasonal pools, the surrounding vegetation. All these components, interacting components of vernal pool ecosystems, they're all affected by climatic conditions. So recognizing that. Climate change, again, one of the leading research interests and concerns in terms of ecology and environment. Lots of research has gone into climate change and has shown that climate change has led to changes in phenology of timing of plants and animals in many ecosystems. Vernal pools have been largely unexplored. What we're interested in doing with this particular project is focus our project on vernal pool phenology. So with our overarching question, we intend to tease apart the importance of local versus regional factors. Also addressing the degree of asynchrony between plants and animals as environmental conditions change and over time. And as you can kind of imagine, that in order to address this type of project, we really need to cast a wide net, get a lot of participants involved in this research project. So we're very excited that you have joined us and are considering participating in our project. And again, also with the EREN model, the importance of sharing our research findings through conference presentations and published manuscripts. Next slide. So what will project participants do? Project participants, if you join this project, which we hope you will monitor the phenology of amphibians and trees and also local environmental variables at small forested pools across time, contributing to our large data set. Dave, I think you can take over now.
Great, thanks, Mary Beth. As Mary Beth mentioned a few slides ago, we are really excited about the diverse backgrounds of folks who are interested in potentially joining the Vernal Pool Phenology project. We have individuals from R1 institutions, R2 institutions, community colleges, high schools, middle schools, non-governmental organizations, governmental agencies, and we also have private citizens who are interested in joining us. and we want you to participate, but you may be wondering, what exactly do you need in order to do so? And so I'd like to take a few minutes right now to run through some of the main things that you would need in order to participate in the project. First and foremost, you need a pool that has breeding wood frogs and or spotted salamanders. So you could have a pool with just wood frogs, that's totally fine. You could have one with just spotted salamanders, totally fine. You can have one with both, great. It doesn't matter. We're interested in both of those wide-ranging species. The pool does need to be forested, and Thelina will touch on that in just a moment. It also needs to be fishless and less than about a fifth of a hectare in size. And the reason we have that size limit is really to make this surveying manageable. Right? So if we get pools that are much larger than that, it's really difficult to be keeping an eye on all of the trees and to get a really good sense of whether egg masses are in the pool or not. You'll also need about $600 or $700 worth of equipment. I don't want to go through this list one by one. I just want to point out the fact that we do have a detailed list that you all have access to in our Google Drive and in the email that was sent out earlier this week. And I'd also like to highlight the fact that many of these items may actually already be in your possession. Many folks have waders or hip boots, maybe binoculars, DBH tape or Taylor's tape. So a lot of this may actually already be sitting in your lab, in your classroom, in your garage, or in your basement. If so, wonderful, you can put it to good use. There are a few items in here that you may need to purchase, and we have URLs actually embedded in the list. So you can go directly to the site where you'd want to purchase those items. So for example, we require 2 hobo pendant temperature and light loggers to be placed at each site. And we also, for anybody interested in wood frogs, if you have wood frogs in your pool, we're looking to put out audio recorders or acoustic recorders at each site, and we plan to use audio moths for those as we move forward. So those are the 2 items that maybe folks need to buy special for this project. I know that not everybody has acoustic recorders. It just so happens that we may have a few extra ones. So if you need an acoustic recorder and you've got everything else on this list, or you can acquire everything else on this list, please let us know. Some of you already have. Others, we're going to give you an option to let us know your needs in the post-meeting survey that Mary Beth mentioned. And once we get a list of everybody who needs equipment, we'll be able to determine who gets those, keeping equity and inclusion at the forefront as we're making those decisions. You'll also need to have the ability to complete twice weekly surveys for 6 weeks for at least a year. These surveys are used to monitor the phenology of plants and our breeding amphibians. You'll also need a little bit of time to set up your site, of course. You'll notice that I said at least a year. This is the pilot year. We're looking to get feedback from everybody and to begin collecting data, but our ultimate goal is actually to run this project for at least 5 more years into the future. We're not asking for that huge commitment right now. We would love everybody who gets into the pilot year to continue on, but at this moment, we're just asking you to give us a year. And as Mary Beth also mentioned, we do have materials developed for every step of this project. This is just an example of the data collection sheet for our phenology surveys. And finally, you need to have a willingness to share data. And I assume everybody in here already does, because you're interested in collaborating, and that's an enormous part of collaboration. So with that, I'll turn it over to Thelina.
Thank you, Dave. So I'm going to go over like the basics of our protocol, what you are expected to do as you join this network. And in the emails that you received from probably from Dave, you already should have access to the protocol in detail that I'm going to quickly go over. So Dave did go into a lot of detail regarding the site selection, the fish list, the size parameters we are looking for. And I do want to emphasize that it can be seasonally flooded, typical vernal pool, or something that is permanent or semi-permanent. That's fine. But it's important that you have some forest association, forest embedded or forest associated with the wetland you are going to monitor. And why? That's because we are looking into the phenological synchrony or asynchrony between pond breeding amphibians and trees, right? So because of that, we require a minimum of at least one dominant woody species where you actually have at least 5 of those individuals within a 5-meter buffer, right? And if you don't have that many within a 5-meter buffer, you will have to get that number of trees, at least for one species, within a 10-meter buffer, right? So if you don't satisfy, you have to satisfy those criteria in order for your pond vernal pool to qualify for the study. And we strongly encourage that you do the site selection in fall for a variety of reasons that I will explain later. And probably, like, in certain areas, depending on where you are from, you still might not have complete leaf off. So you could do tree identification and count the number of trees around easily right now in this particular in this season before the first snow. Dave, next slide.
Belina, before we move to the next slide, I realize that it may be a good point to press pause for a moment.
Oh, good
See if
point.
anybody has any questions about anything that you need to participate, including questions about the characteristics related to your potential site, or if you have any questions about anything that Mary Beth talked about, please ask those now and
good point.
hopefully get answers for you.
I do have a question, whether it's okay to include a site that is being actively managed where invasive shrubs that might be surrounding a vernal pool are being removed throughout the season.
We haven't discussed this in depth, or actually really
We
at all
haven't.
in our leadership group. My initial feeling is that as long as none of the trees, larger trees, are being cut down, then that may be okay. But I'll let anybody else jump in here because they may see a problem with that I'm not.
Yeah, if it is actually from what I'm hearing, it is invasive shrubs that are removed, not larger trees. So that dominant tree species around the 5-meter or 10-meter buffer around the vernal pool, those are not going to be cut down or removed unless some of those trees are Ailanthus and Polonia. So I wouldn't, if it is shrubs, I suspect it should be fine, but I believe the leadership team should revisit it. But if it is trees that are going to be managed, harvested, it could be a problem because your dominant membership within the community is going to change, not by natural death or natural recruitment, but by human management. So any other questions that we can address, or any follow-up regarding the management part?
I might talk to you offline about it because our study sites are oak management sites where they're actually trying to create some active canopy gaps as well as remove invasives. And there has been in the past some girdling immediately around the vernal pools that have affected largely silver maple that encroach, but they're leaving certain other species. So if I can get a list, in advance of which trees will remain and know that I have at least 5, it should still be generally okay, I think. But I could follow up with you later.
Absolutely, absolutely.
And I'll just add something, just that we are, as I think you probably realize, to address the questions that we want to address, we really do need a large buy-in, a large number of people participating, lots of sites. So, whether or not down the road, we might have to throw out for a particular analysis, a particular site for a part of analysis, that would be a decision down the road anyways. I think right now we're trying to cast a large net, in my opinion.
Right. Any other questions? If not, Dave, we can move on to the next slide.
Selena, I just thought I would voice, in case people aren't looking at the chat, a question that came up in the chat just for everyone to hear, which was, if you're only monitoring for spotted salamanders, do we need to have the audio mods? And of course, the answer is no. And so, we list that as our equipment. But yeah, if your site only has spotted salamanders, then no, you don't need to invest in that. We were having a bit of a conversation in the chat about, if you have spotted salamanders and wood frogs, but you don't have the equipment in a given year, I mean, we can talk about this as a team as well. However, my gut reaction to it was, yeah, if you can only monitor spotted salamanders in the first year when you don't have audio mods, fine. And then hopefully in the future, we can get some equipment to people, or you can acquire equipment. So we can get back to people about that, but we just wanted to address that.
That's great, Jen, and I agree with everything that you just said. I also do want to note that it's difficult, or it may be difficult to acquire audio moths before next spring if you haven't already ordered them. And so for this first year, if you have access to another acoustic recorder, if someone else in your institution or organization has one lying around that isn't going to be used for that 6-week period or 8-week period, then go ahead and use that this year. They should actually all function the same way. Ideally, we'd have everyone using the same acoustic recorder, but for this first year, due to all of the chip shortages and other challenges, supply chain challenges, it may be necessary for some of us to use something like a Swift or a different acoustic recorder, and that's totally fine for especially year one.
I was just wondering, when looking at the AudioMoth website, they're only available to purchase in 10 packs. Is there going to be like a group buy-in where if we get like 10 people together that want to buy it, that we could all purchase it together and organize that? And is that something that you guys will be organizing?
So we hadn't talked about organizing that. We are aware that, I mean, Eva, you and I were speaking in the chat a little bit about this, but we are aware that at one point they started doing the 10-pack offer. I hadn't realized that until you mentioned it, that they had stopped allowing you to do the single purchase. We're hoping that down the line, we might be able to, we're going to be working on some small grants, we hope, to try to get equipment grants to make some of this equipment more broadly available, to encourage more people to participate. I mean, I don't know, I guess we'll have to talk about whether we're going to organize that, but I think that we can absolutely facilitate like a place where people could get together and organize that.
At least like a common spreadsheet where you sign up.
Exactly.
Where you have adequate purchase power. So at least we can initiate something like that.
Right.
And that'll be part of the survey questions for next week too.
Yeah.
And my hope is from following these audio moths over the last year and a half or so, my hope is that maybe starting in January, they become available again for individual purchase, but we'll keep an eye. It's a good question.
Thank you so much.
All right. Right, let's continue on if there aren't any further questions. So now let's talk about very briefly what we are going to measure, right? We are looking into, we plan to measure the hydrologic variability in the pond, in the vernal pool, the canopy cover, and air and water temperature. So in order to measure the hydrologic variability, we are going to use one of the most dead cheap setup, like a homemade water gauge. If you look at the image in the slide, what you're looking at over, yeah. Dave, would you mind running your mouse on it? Yeah, so what Dave is showing, that is actually a PVC pipe, a half-inch diameter PVC pipe calibrated by Sharpies and electric tapes, right, to measure the water depths, and that is sort of we first ground a rebar, and then we put the PVC pipe on the top of that, and then we screw tied that PVC pipe to the rebar to stabilize it, and then it actually worked pretty well, surprisingly well. It stays like that. It doesn't move around, and then this water gauge, we can use it to set up the hobos. So we earlier mentioned we want to measure both air and water temperature. So you have to have 2 hobo units. Dave mentioned that very briefly, and you need to have one of them on the top of the water gauge, hooked to the top of the water gauge, and another suspended about 10 centimeters below the water surface. And in our protocol, we describe how you can do that, like, with a float plus some weight, which actually weight helps to suspend it 10 centimeters beneath the water surface, and the float will prevent it from going further deeper in the water column. So, and then, so we want you to set this up possibly early in the season. It's not a bad idea when you are site selecting. If you select the site, if we want the water gauge to be installed in the deepest point. So that's again why a fall setup, fall site selection is a better idea. You can easily see the bottom of the pool and set it up. If your pond still has water when you set it up, not the end of the world. Whenever it dries up, you can move it towards the deepest point. And we provide some instructions how to recalibrate your measurements if you had to shift the deepest point because your pond happened not to dry out completely. So, and I'm losing where I was before finishing up. Okay, so and so you can set up the data loggers when you set up the water gauge and leave it there. And when you launch it first before deployment, with the fresh batteries, those data loggers will actually work for over a year without any problem. And we recommend that once you finish surveying, a month after, to remove them from the ponds, right? Both of them, remove them from the ponds and then put them into the data shuttles and extract the data, right? And canopy cover, now we want you to measure canopy cover only twice in a given calendar year. First, you measure it leaf off. That's going to be late fall. Like in my case, I do it in late fall. It's fine to do it early winter or early spring. And then the second one, midsummer, right? So based on, where you are, time when you actually have the complete full leaf out and complete leaf off. So how do you do that? Well, again, we use smartphones, right? So there are several, we read several procedures where you can use the smartphone photograph to actually quantify and analyze the canopy cover, which actually, regardless of the smartphone, iPhone, Android, Pixel, doesn't matter. Actually, some of the studies have shown consistent conservative values, even if you use different phones with different magnifications. And the optional part here is, one more thing about canopy cover, make sure that you are using a tripod or some sort of a way to stabilize your phone with a leveler, so that, the canopy cover measurement is very sensitive to you holding the camera straight up, right? So it's really important that you worry about the stability of the phone as you take the photographs, right? And also the height, the proper height. We have described all of those in our protocol. Now, optional one is to set up a trail camera for two purposes. So earlier I mentioned about setting up a water gauge. Now, how frequently do you read water levels out of the water gauge? For 6 weeks, twice a week. That's the minimum we require, right? And then during those twice a week, you do several other things, which we will come to later on. So one of the things you measure is you read out from the water gauge. Now, depending on how far your deepest point is, you might need a pair of binoculars to read the water level. Or you can actually use your phone again to like zoom in and read the actual water level. So in addition to that, if we need finer level data about water levels, like daily data, what we recommend is set up a trail camera right in front of the water gauge, and then program your trail camera to do like a scan shot once in every day, right? And we would also recommend setting up another trail camera aiming at the vernal pool, where we can photograph what wildlife is accessing that, right, during the sampling season. So remember, trail camera is optional. We have provided some links and recommendation for trail camera type, right? Again, that's optional. Next slide.
Selena, before you move on, can you go back one slide, Dave? I don't know if I heard you say it, and I thought it was worth mentioning that one of the reasons why we're using hobo data loggers to do water and air temperature in situ instead of using a nearby weather station to have ambient temperature, for instance, is because then we're able to know when the pool dries by looking at when the water and air temperatures kind of stabilize and become the same.
Similar.
Yeah, so it's a really nice, that's one of the reasons why we're doing that, even though some of us have weather stations really close by. So yeah, just wanted to mention that. So we can monitor hydro period.
And we really can piggyback from this finer resolution temporal data in this case, right? So yeah, moving on to the actual phenological measurements. So when do you start? Well, you need to have some knowledge about your locale, right? And we have recommended in the protocol for amphibians, when do wood frogs start calling in your locality? Well, there is a nice citizen science framework called FrogWatch. You can log into FrogWatch and then follow up like in your area when people have started recording, have noticed emergence of wood frogs. Normally, if you have some connections with the local zoos, you tend to actually do this kind of work for outreach activities, so they can help you figure out when exactly on average wood frogs start to call. So we recommend that you actually start making these observations a week ahead of the historical frog calling phenofaces. And then from that starting point, we want you to actually keep monitoring for 6 weeks, 6 consecutive weeks. In each week, we want you to visit the vernal pool twice, right? And let me talk about the amphibian phenology part first, and then talk about the plant phenology component. So amphibians, like Dave mentioned earlier, we are looking at 2 species, spotted salamanders and wood frogs, right? And in the image below, we are actually showing you the 3 phenologically relevant reproductive events we are looking at. Whereas Dave's pointing, that's wood frog egg mass, and then you are looking at spotted salamander egg mass, and then a spermatophore, right? So we want you guys to walk around the pond periphery and wade in the periphery and look for this evidence. So you need to, it will take some training initially if you haven't done this before, to locate them, particularly like wood frog egg masses. When they are submerged, it's hard to see directly, so you need to first actually train your eyes to see where things are. Spermatophores, actually, they deposit on the bottom, and some of you already have probably know this. So there will be some initial training to get your eyes trained for that particular locality. And we have some of the teaching material actually provide detailed descriptions about what does these 2 species egg mass look like, right? Where to expect them? Are they, none of them are in deep water, right? They're actually closer to the surface. So detectability is fairly high in vernal pond periphery. So, and then what you need to do is simply record, starting from that whatever the first week you start to go there, every 2 weeks visit the vernal pool and record when it appears. If you don't see it, record that you don't see it. When you start seeing it, you stop making observations for that particular phenophase anymore, right? So once you cover all 3 within the first 6 weeks, whenever you stop, so do the first observation, you cease making continuous observation for the same phenophase. Audiomoth, we have already spoken about that, so I will not go over it. Just to clarify, it is only when you have wood frogs in the vernal pool that you are monitoring. Now to the plant phenophase. So like I mentioned previously, we should be looking at up to 5 codominant species in the vernal pool periphery. 5 meters, if you actually can find minimum of 5 individuals of a single species. If you can't, you go up to 10, right? And I said up to 5 codominant, not minimum of 5. So then you document 3 phenophases: bud breaking, leaf bud breaking. Like you can see, like that's when you actually see the leaf emerging from the bud, and then complete leaf emergence when you see the leaf from tip to the petiole, and then third is flowering. Right, so those are the 3 phenophases we are looking at. And remember, we are trying to apply these phenophases to, well, at least the first 2 phenophases are applicable to both hardwood and coniferous, right? I think that's all from me. Any questions regarding the protocol? Jen, you are monitoring the chat line. Any relevant questions that we need to bring up?
No, there are no questions at this point.
Okay, in that case, Jen, it's you.
Okay. I'll try to just talk about this kind of quickly. You have a bunch of educational resources available at the link that we shared earlier in the chat. So Mary Beth told you a little bit about EREN, Ecological Research as Education Network. And so a lot of us end up incorporating projects like this one into our teaching. And I think many of the participants in our first pilot season last year used these protocols with either independent study students and or courses. And so this on this slide is just a list of some of the courses that we have already incorporated this project into, just to show you an example of kind of the range of courses that how adaptable this project is and how many kind of different concepts you can link this to. And so it can really show up in a lot of places where you're doing education. And that is true whether it's formal education at the university level, at the high school level, whether it's more of an environmental ed, community education, or independent research, K-12, whatever. We do want to say that while we are really interested in sharing this with our students and having our students be really involved in this, and that's been really a great experience for us. And I'll speak for me and Dave because we work closely together at UNH, and we have some of our students are on this call tonight who have participated in this. It's been really, really awesome to have huge groups of people out there working on this project. And but you do not need to incorporate this into education, to teaching. You do not need to use the teaching materials, or you can just do this on your own as a research project. With an independent study student, it doesn't need to be incorporated into teaching. One thing that we will do every year, and we did this just kind of at our institution last year, is have an annual kickoff meeting. So this will be Zoom like this, where we'll just do a brief overview of like who's participating and some of the central questions, some reminders about I don't know, tips and suggestions and reminders for protocol. And we'll do that every year so that you can invite your students, or you can come every year to that meeting. And that was a really great experience this past year, to have students from multiple courses independent studies coming together and realizing that they were all a part of something much bigger than themselves and then their class. So we look forward to doing that every year. Can go ahead. So I was looking at my slides over there. I'm like, it didn't change. This is just the overview sheet of our instructor guide. So in that link that we shared with you earlier, there is a teaching resources folder, and within that we have instructor resources and then student handouts, and these are really just our initial kind of prototypes of these teaching materials from what we did last year. And we are planning to this summer develop even more materials for the coming years. But we wanted to have something for this first year if any of you want to incorporate this into your teaching. So this is just an overview of the instructor guide. We've developed a couple of labs, one that you can actually have a lab experience where you're setting up your site, if you can do that early enough in the year or in the fall. And then a lab where you can go out and actually do the phenophase surveys. And we also have pre-lab materials to have the students read a little bit of literature about the issues explored here. And a bunch of other materials. We also have examples of the types of resources we gave our students last year, how to identify different egg masses like Thelina was talking about earlier, how to identify key phenophases on the plants that are dominant in our local area. So you would have to adapt that for your local area based on what your species are. But we are we welcome any suggestions too if people are looking for a certain type of educational resource. We're happy to work on developing that with you. So can go ahead to the next slide, please, Dave. So I also wanted to mention, and kind of did already, with how many different types of education and types of courses and undergraduate research that you can kind of center around this project, that we have already found this such a benefit to have this baseline kind of environmental data about these sites. And we have already started to add on new kind of side projects at these sites. And so, yeah, trail cameras have been a really fun part of that. The students who are out working on tree phenophases and looking for egg masses also really enjoy seeing some of the wildlife that are frequenting these sites that we work at. We were just out this morning doing some forest inventory practices, like trying to develop skills around how to do a forest inventory, a tree inventory around some of our vernal pools. And of course, if you're collecting acoustic recordings, you could ask more questions about acoustic ecology since you're already gathering those data. Those are just a couple of examples, but there is so much that we can do with these sites once we get a little bit of baseline data. And then next, and I think the next slide, I mean, Thelina really talked a little bit about these stages and what you need to do, but we wanted to just kind of show you this visual of some of the benchmark moments, the major project activities that'll happen and when they happen throughout the year. Of course, hard to say when you're going to be setting up your site. You might decide to set it up in the fall or the winter or very early in the spring. And then once that's done, and all you need to do each year is kind of reprogram your hobos, check your batteries, but that part's done. And then annually after that, you're really limiting everything at that point to kind of a 6 to 8 week window where you're going out and doing the surveys, taking those photos that Thelina mentioned. We don't have that on here, and entering and submitting data. So I don't think we need to belabor that. Can you go forward?
Yes, definitely. And just a reminder that the short survey will be.
Yeah. Yep. Oh yeah, and so we wanted to show you, this is kind of exciting. Mary Beth showed a map earlier of all of the EREN participating institutions. This is only those of you who have responded and said that you're very interested in being involved in the Vernal Pool project in particular. So we have had really overwhelming response, and you can see this fantastic geographic range that we're going to hopefully be able to cover. And so we're really excited about that. I'm really happy that so many of you are able to be here tonight. We want to just answer questions at this point. So yeah, is there anything, Mary Beth, in the chat that we should address?
Yeah, there's a couple, somebody asked if there was going to be details on how to make the staff gauge, if that is going to be available. We do have it explained in the detailed protocols. We've got tons of photos of these and on how to make them. So we're going to be also adding over the next couple of months detailed guides that actually show, images and even possibly little mini videos to help with much of the work that we're doing here. That was one question. Anybody else want to add anything on that? Because I do have another question I'll ask.
I suppose I just want to say that if you are reading through the protocols and you're confused about something or you're not quite sure, you also can always reach out to us, and we'll be there to clarify and to guide you through it, and then also to make any tweaks to the protocol that we need to make things a little smoother for folks moving forward.
Dave, I was just noticing a question in the chat about like frequency of sound recordings and the length of time that we'll be doing sound recordings. I would say the sound recordings are a bit longer than 6 weeks because we want to put them out a little bit earlier so that we actually earlier than we expect to hear wood frogs, so that we actually get those first calls. So they'll go out a little bit earlier. Our experience last year was that we didn't have to replace SD cards or anything like that. The files were pretty small, and so you could just leave it out. Batteries were fine, just leave it out that entire time. We're currently working on figuring out exactly, we collected a lot of data last year about like basically 24 hours a day of sound recordings to try to figure out exactly the answer to your question, which is when should we be recording throughout the day and night, and for how long? And so we're going to provide that absolutely before spring.
Yep,
Dave, I don't know if you wanted to add anything to that.
That's perfect. I guess I will just say, even if you recorded for extremely long periods of time every day, you still won't need to replace your SD card or batteries. And you already mentioned that, Jen, but we'll be getting more specific answers to those questions in the coming weeks.
So that was the other question in the chat that I was going to mention. So I think if anybody has other questions and they just want to unmute themselves and ask, feel free to do that.
Mary Beth, do you see the question in the chat?
Yes. Will the local EREN affiliated colleges be made aware of participants, possibly of their engagement support? I guess I'm not sure the question, what that means. Jen, do you know?
I'm not exactly sure. We have sent out our kind of recruitment call, like our welcome to everyone in the EREN network. We can, I suppose, also let the EREN members know who's planning to participate in the first year in case someone nearby is working on it, so that you could try to collaborate, if that's what the question was. But if you could clarify so we make sure we're actually answering the right question, that would be great.
Sure. We've worked with some other students, Franklin and Marshall College, for example, on water quality issues. And I guess my question is if some of these schools have people that are interested in helping us with doing this, would probably be beneficial to all, because they're going to potentially bring some stuff and we can help them learn from our experience as well.
And did you say that you already have contacts at these schools?
Well, I also do a lot of, we also do a lot of water quality management and supporting the cleanup of the Chesapeake Bay. And so as part of that, some of the people that have been drawn into to form teams to brainstorm how to work stuff. We've had some of the young college folks engaged. So not specifically on the vernal pool stuff, but it was great to get them involved because they can bring a little bit more modern science to us than some of us old guys have, you know? So, so that was, I guess if there's any of them that are interested in this type of stuff, you know, there might be some ability for a relationship to be formed there. And then,
Yeah, I mean, we encourage that in any way that we can be helpful in connecting you or anyone with, you know, EREN affiliated faculty. We'll be happy to do that. Yeah. And Mary Beth, you said, you're answering a different question on the recording? Is that what you're saying? I just saw your chat.
Yeah, I was gonna, are we done with this question? Should we move to the next one?
Sure, I wasn't sure if you were responding to that question. That's why I was asking. Go ahead.
Different one. Yeah, so this was about the authorship guidelines. So let me look at that question. What level of collaboration and co-authorship will be expected of project participants beyond collecting, sharing data? So there, I was just looking up what we put in our proposal. I thought we had more details. I'm looking at our original proposal for what, so because this is an ecological research education network project, we follow the EREN's authorship policy, and that is available on their website. I'm going to put that into the, I think if that link works, it should show that. We will be, what we also do as part of these projects is that we also track contributions over time. And so there'll be, if you're running, you know, if you're running a site and contributing data, and we have, I think in our policy, we had, it was a minimum of 1 year's worth of data, then you would be contributing data for the project and qualify for authorship on research publications. As you're probably aware, as time goes on, there's a lot to do with data analysis, all sorts of phases of bringing raw data into a research publication, contributing to writing, commenting, etc. So we track contributions over time as a way of, in terms of gauging like authorship order, etc. And lastly, I'll just mention that EREN, you know, understands the importance of contributing and getting acknowledged and having contributions of researchers on publications. So we take that very seriously. We are evaluating currently, and I think Jen's on the subcommittee of revisiting our authorship guidelines to make sure that to make any fine-tune changes to that if needed as we all progress as collaborative research scientists. Jen, do you have anything to add? Because you're on that committee.
No, we're really kind of in early stages right now. So,
Okay.
but your response, it was great.
Okay. And so if you have any further questions, feel free to shoot an email to us and we can chat more in details about those things.
I saw another question come in while we were talking about authorship about NPN, National Phenology Network. And, you know, do we have any plans to either share data with National Phenology Network or to use their built-in platform? We met with some representatives to talk about National Phenology Network and how we could utilize some of those data or at least use it for, I don't know, kind of a reference or point of comparison last summer. And I would say that part is still kind of under development. So we're still trying to figure out how we can incorporate some of those big data sets into the data we're collecting in the field. But yeah, it's absolutely on our radar. It was kind of part of the initial intention was to try to figure out how we could do that. So to be continued. We'll update you soon. Does someone else, I feel like I've been talking a lot. Does someone else want to address this great question, which I love, about what are our hypotheses about connections between amphibian breeding and tree phenology? Some of those potential mechanisms or, mechanistic connections. Like, why do we care about asynchrony, I think, is the question. Anyone want to address that? No?
Yeah, I think if I were to mention, and please do jump in. So this idea started with one of the papers we read from Central Europe, where for a smaller area they were trying to understand phenophases between different types of tree functional groups and with amphibians with different reproductive strategies. They were trying to look into this phenological asynchrony between different phenophases of this distant taxa. So in from a very basic science perspective, like if it is the climate that is driving all those phenologies, regardless of the phylogenetic affinities between these taxa, right? There should be some sort of a conservative gap between the taxa in terms of when these phenophases emerge. But with climate change, with time, or with geographic differences, how much this asynchrony is going to shift, right? Is it going to shift temporally with respect to climate change? If it does, is it going to shift towards the same extent, to the same magnitude? And how does that shift spatially? So those are the kind of questions we are trying to raise in this particular project. And the mechanistic relationship behind it is because, like, there are several mechanisms we actually talked about, at least among the leadership. Some of them, one of them is, the canopy cover, right? If you actually have an early leafing out around a vernal pool, that is going to create a problem for the vernal pool's hydro period. It will, with the evapotranspiration kicking in, the vernal pool will decline in its hydrology, right? Or like go to a shorter hydro period. That is likely to trigger, an early phenophase of amphibians, just to keep up with the shifting phenophases within the community, right? That's actually one mechanistic relationship, and there are other mechanistic relationships with respect to secondary metabolites a plant could produce as the growing season starts. They could release certain secondary metabolites through defoliation or through roots that will impact the water quality, and that is going to impact the growth and metamorphosis of amphibians. So those are some mechanistic relationships that we can think of. And the paper that we read, they actually found relationship, like very distinct relationships between certain plant functional groups and amphibian phenophases in terms of migration onset and fruiting, like really weird relationships that you wouldn't anticipate otherwise.
And I'll just add a couple things, because I was actually going to mention a little bit about why looking at vernal pools and the phenology of, pools themselves, the fauna, as well as the surrounding vegetation, in particular the trees. And I forgot to mention that when I did my part. But so what we're trying to do, and why we're trying to cast this wide net, is because we want to be able to try to tease apart what are potentially important factors. So it's really exploratory in that respect. So local versus regional climatic conditions. So on site, if your site, for example, if your site is sheltered, water temperature, the water level fluctuations, there's a lot of different things that we can potentially feed into our exploratory model to find out what is most important. But basically, and that's why we want a lot of people participating, because this is the only way we're going to be able to tease apart these things. But looking apart the importance of local versus regional factors, what ones come out to be potentially most important, most interesting to do follow-up studies. And then, as Selena was talking about, was that, the asynchrony or the synchrony between the plants and the arrival of the pool breeding amphibians, that when they initially begin to breed, initially you've got, our different tree species tracking different aspects of climate. So they're budding out earlier. And then once they bud out earlier, then they begin to draw excess water because they're into their growth phase. So then that can affect the hydrology of the pools. So there's a lot of different things that we can potentially tease apart by doing this. I hope.
And I also want to add that there are lots of phenological synchrony, asynchronous studies on plants, a lot. And most of those studies concentrate on a particular species and compare multiple populations or compare different species, but phylogenetically they are within closer distance, right? So why not expand that into two community members that are phylogenetically quite further apart, right? Because the phenological theory regarding asynchrony should still stand because phenology is driven by climate, right? So I think there's a novelty in basic sciences when you are looking at these two members that are, like coming from two different kingdoms.
So I just noticed that it's 8:01, and I think we were planning on shooting for an hour. I think some of us at least are able to hang out if anyone has questions, wants to ask us anything one-on-one quickly at the end. But we want to respect your time. So, right, are there any last things we want to say before we close? Group? No?
No, we do have one final question from Yinging though, about whether or not we expect to use precipitation data from weather stations in data analysis. And I will briefly say, yes, we are talking about different ways that we can pull in data sets in order to look at regional versus local effects of these variables on phenology. Yeah,
we've also considered just talked about doing some kind of snowpack monitoring, maybe by something simple, kind of like our water gauge, and having essentially a snow depth gauge at sites locally to see, if that affects wood frog timing in particular. So yeah, we're thinking about those kinds of things, like Dave said, at the local and kind of regional scale. It's great. But yeah, thank you. If some of you have to leave, we just want to thank you for coming tonight. And we'll hang on for a few minutes and see if anyone has any more questions. But we're really excited about this map and how many people might be involved in the Vernal Pool project. So
yeah, thank you all so much. And please do not hesitate to send an email with any questions. We are so excited that you folks are potentially interested in working with us, and we're happy to get answers to anything you've got.
And look forward to our survey that'll be coming out early next week.